Hello everyone, welcome back, and we are continuing with meiosis. So we will see today we will discuss about meiosis in detail. Okay, as much as possible we'll complete meiosis today. So we are going to talk about meiosis. Okay, so I hope the other things like mitosis we have done and the other things, I think it's clear for you people. There is no need of again explanation or anything else. If any doubts, again you can ask me. So coming to meiosis. So meiosis is not like mitosis, you know that also, you know the difference of meiosis and mitosis from class 1. I have started with that only. So it's easy for you people to understand meiosis very easily. Okay, remove all the confusions first only, be cool and calm, and then listen to this so that easily you will understand. You people know very well that I told you for to understand meiosis, the blueprint of meiosis and mitosis you should know. Then only you'll understand it better. So the blueprint goes like this, that meiosis happens in meiocytes. So, or you should remember the blueprint easily. That means, for example, this is the cell, okay, and it has, let us take easy example, 4 chromosomes. The parent cell is having 4 chromosomes, then it will divide. The first meiosis, it will form 2 cells. What will be the number of chromosomes? 2 and 2. This is called first meiosis. And the next meiosis, what will happen? Second meiosis, it will lead to completely formation of 4 cells wherein we will have 2, 2, 2 and 2. So this is what we are going to study in detail, right? And this is called meiosis 2. So by this blueprint, if you take, for example, if the parent cell is 46, like if you take example of human beings, here the chromosomes will be 46. Then after first meiosis, 2 cells are formed. Then what will be the number of chromosomes there? It is halved. That means the number of chromosomes will be 23 and 23. And what about the 4 gametes which are produced? All the gametes will have, all the 4 gametes will have 23, 23, 23 and 23. Clear? So this you should know very well. That means meiosis is divided into what? It is divided into 2 phases. That is meiosis 1 and meiosis 2. So first of all, to understand it still more better, we will see the complete chart. Okay. So meiosis is divided into meiosis 1. Let us consider this is meiosis 1. Okay. And then this is called meiosis 2. Meiosis 2. Okay. Now you know that mitosis or meiosis, anything, cell is cell only. Whether it is undergoing meiosis or mitosis, cell is cell only. It will have nucleus, it will have cytoplasm, it will have nucleoplasm, and the division process also is same. Slight difference is there between mitosis and meiosis. So first what needs to happen? The nucleus should divide. Already we have discussed this. The nucleus division is called karyokinesis, and after the nucleus divides, then what will divide is the cytoplasm. So first will be the karyokinesis process, and then what will happen is the cytokinesis. So in meiosis 1 also we have both karyokinesis and cytokinesis, and meiosis 2 also we have karyokinesis and cytokinesis. So let us consider meiosis 1, okay, and it is divided into 2. That is, this is first is karyokinesis and this is cytokinesis. Clear? And here also we have, again in meiosis 2 also it is divided into 2. First one is again karyokinesis and again it is cytokinesis. But since it is, the terms are same, then we should understand it, no? You should not get confused. So we will name it as karyokinesis 1, cytokinesis 1, because karyokinesis and cytokinesis which is happening here is related to meiosis 1. So it is 1 and 1. And what will be here? Obviously it is karyokinesis 2, cytokinesis 2. Okay, in order to differentiate. Now, what are the stages in karyokinesis of mitosis? I have already told you that whenever mitosis is happening, in karyokinesis, it's not an easy thing. Comparative to karyokinesis and cytokinesis, cytokinesis is a little bit easy because the major things are already done. Only the cytoplasm should be divided. It is easy. Karyokinesis is not that easy. Why? because chromosomes should be formed. The DNA will be there, DNA should condense to form chromosome, and the centriole should start forming the spindle fiber. Chromosome should be kept at the center, chromatid should be separated. So many things are there, right? So karyokinesis takes for a long time, and there are again different stages. So the same stages are there here also. Let us see which are the stages of karyokinesis. 1. We have what? First one is prophase. To understand, it is prophase 1, metaphase 1, anaphase 1, and then telophase 1. Clear? And after these all, what will happen is, it leads to cytokinesis 1. Once the cytokinesis 1 will happen, then this is called meiosis 1, wherein from one parent cell, how many cells are produced? 2. Okay, that is clear. But what is the number of chromosome? Reduced. We were talking about the blueprint. We have taken, for this example, we have taken parent cell has 4 chromosomes. Then here, as it completes meiosis 1, what will be the number of chromosome in the daughter cells? It will be 2 and 2. It is reduction division. And I'll write that also. Meiosis 1 is always reduction division. And meiosis 2 is equational division. Equational division. So see here. So again, what about karyokinesis 2? Again, if you see, in karyokinesis 2, again we have prophase, metaphase, anaphase and telophase. But how to remember? Again it is what? Here, to remember, what we need to remember here is, it is 2, 2, 2 and 2. Prophase 2, metaphase 2, anaphase 2 and telophase 2. So, after this, so here already one cell in this meiosis 1, one cell, I will show that also here to make it clear. So, for example, we had taken cell with 4 chromosomes. It divides and forms 2 cells. And what will be the number of chromosomes? 2 and 2. So, this is meiosis 1. Then what will happen? These 2 will enter meiosis 2. So, already there are 2 cells which are having 2 and 2. What will happen now to these 2 cells here? As they complete karyokinesis and cytokinesis, they will form totally how many cells? They will form totally 4 cells. And what will be the number of chromosome? 2, 2, 2 and 2. Right? So please take down in this manner only. Okay. So this is along with this diagram, and if you want me to show the difference. Right? So this is meiosis 1, which is reduction division. You can see 4 has reduced to 2. Meiosis 2 is equational division. So these 2 cells only I am showing here. Okay. So they have produced 4 cells. All the chromosome number are, they are same as the parent cells. So this is the what you can say. Overall, you should have this idea about meiosis. Okay. Karyokinesis 1. In that we have prophase 1, metaphase, anaphase, telophase 1. Then it goes for cytokinesis. Then only 2 cells are formed. Then those 2 cells will undergo meiosis 2, wherein again it starts with karyokinesis 2. And prophase 2, metaphase 2, anaphase 2, telophase 2, and then it will go forward. After it completes, like how it completed all this thing and then it went to cytokinesis, in the same way, after completing all these stages, it will enter cytokinesis 2. Here it is cytokinesis 1 and here it is cytokinesis 2. Once the cytokinesis 2 is completed, how many cells are formed? Totally 4 cells are formed. Clear? So this is the overview of meiosis 2, and this chart is very much important. Please take down this much. Okay. Now, as I have told you, okay, see here. First of all, what we need to understand here is, comparative to mitosis and meiosis, meiosis is complicated. Why it is complicated? Because there are 2 important concepts what I have already discussed with you in the previous video. I told you about homologous pair and I told you about crossing over and recombination. Because of these two things only, meiosis is very much different than mitosis. So where actually that happens, where it takes place, so that I will give you an idea here. Remember, meiosis 2 is very easy. It is exactly like mitosis. It is almost similar to mitosis. No change at all. It is almost similar to mitosis. But meiosis 1 is not like mitosis. Meiosis 1 is very much different. Why? Because, now I'll be showing you why it is different. I'll rub this, okay? I hope you have taken down properly. Fine. So, now what you will do is, you cannot rub like this. So, what you will do is, when you are writing only, you take a lot of space and then you write, okay? You take a big page and then you write this overview. Then only we will start in detail. So, we know this much. Now, why this plays very important role, meiosis 1? Because in meiosis 1, this prophase what we have, the prophase 1 of karyokinesis 1 of meiosis 1 is very much important. What happens here is, I'll be showing it here. Prophase 1 is divided into 5 stages again. Prophase 1 is divided into 5 stages again. Okay, which are those 5 stages? Let us see now. Okay, the stages are like this. Try to understand the phases. The first one, I am talking about meiosis 1, karyokinesis 1, in that prophase 1. I am not talking about this. Prophase 1 is a very long duration. It will take very long time because it is again divided into 5 stages. And these 5 stages only are important for recombination and for the variation. Okay. So which are the 5 stages? We are going to talk everything in detail later on, but we will just understand the overview here. The first one is leptotene. Second one is zygotene. Third one is pachytene. Okay. Fourth one is diplotene. And the last one is diakinesis. Diakinesis, whatever. Students will have problem with pronunciation also. See, however you want, you can remember. Ultimately, you should just know the spellings properly and you should just know what is happening there in that concept. If that you know, it's great. Leptotene, zygotene, pachytene, whatever. Diplotene and diakinesis. So these are the 5 stages which phase? Of prophase 1 of karyokinesis 1 of meiosis 1. Clear? This is how it is different than mitosis. This is the overview. You have to take this chart. We will be discussing everything in detail. We will discuss meiosis 1 first. We will discuss this thing first. Then we will go for metaphase, anaphase 1, telophase 1. Then we will go for meiosis 2 also. Okay. Take down this much.